did. And I'm really glad to be here tonight and talk about something that I think is really fun and wonderful, and that's, you've already observed, plant colors. Some of the plants on there are wild native plants, but one of the nice things about this whole topic is you don't have to go to the wilds, and it doesn't have to be just natives. You can watch plants be colorful and think about why and how they do it in almost any environment that has any plants, and I'm sure you have plenty around here. So it's fun to put something like this together. I hope you find that it's fun and enjoyable.
I can't hear.
You can't hear?
No.
How about that? Kevin said about taking a boring subject and make it interesting. I hope I don't take an interesting subject and make it boring. I don't know how much louder I can get. I'll try. The microphone's here. I don't have an extra hand for it. But this is the subject for tonight. I hope you learned something. And so, which one am I? There we are. Where's the page now? There it is. Is it on there? Oh, there we go. Thank you. One of the finest books on the subject, if you're interested at all, is this one, Nature's Palette, The Science of Plant Color by David Lee. We bought it as a hardback several years ago. It's now available on Amazon for $20. It's really an excellent book, full of color illustrations, and I don't think you'd be disappointed at all if you had that as a reference for yourself. And I'll take one quote, or a near quote from it, that David Lee says, "Knowing a little about the science of plant color makes the phenomenon even more rich, more elegant." And that's how I feel about it. And if I were to come up here and not try and explain some of that, I'd say something as short as, well, plants are different colors, and that's because they contain different pigments. Isn't that nice? But you deserve a little more than that. So I do want to go into the science of how it works, and a little bit into the ecology of what plants get in the deal. And in doing that, we're going to go into a bit of detail. If the detail suits you, that's terrific. If you find it a little bit less than interesting, that's fine. Persevere a little bit. The more color and the less science comes toward the end, so if you endure what's up front, you'll be in for a better show later on. And there's no quiz, so you can just enjoy as much of it as you want and wait if it's a little more detailed than you'd like. But I think it gives the entire subject a great deal more richness and appeal and interest. So we start out talking about plant colors by talking about how we as humans perceive color. And color is very much a construct of our brains and our visual systems. Here's an illustration similar to one that we'll use a number of times tonight, and that is to just illustrate the spectrum of white light from the purple end through green and yellow to the red end. And across the bottom here is the wavelength, the distance between the little wiggles of the light waves in nanometers. And up the side is, in this case, the sensitivity of the color receptors in your eye. So we'll see this again and again, and this is just the visual spectrum. If you took white light and spread it out by wavelength, just as Isaac Newton did many years ago with a prism and investigated the nature of color, this is what you'd see, from violet to red. Beyond that is the infrared. We don't see in infrared very well. And up here is the ultraviolet, and we don't see ultraviolet very well. Basically, blue wiggles are fairly close together, and red ones are a little further apart. And you have in your eyes 3 different kinds of color receptors, 3 different cones, and they each have their own sensitivity to particular colors, meaning the wiggles. This is one whose sensitivity is maximum here in the blue-violet end of things, and these two are pretty close together. This one has its sensitivity right between green and yellow, and this one more in the green, but they respond to a range of colors. The fact that this line slopes down means that if you show this cone a little bit of red light, you'll get some response. It just won't be its maximum response. And what we perceive as color is based on the balance of outputs from these cones and these cones and these cones as we view a given scene. And your brain interprets that as whatever color you're going to see. Now, we could stimulate those with a single color. I could use this red laser pointer. This red laser pointer is a single red wavelength, a very pure wavelength. and it might stimulate this cone down here, and it would signal to the brain that it's seeing red. But you can also see red, and in fact, most of the time you're not seeing a pure color at all. You're seeing colors across the spectrum in varying degrees, varying intensities, and they're exciting their cones to varying degrees depending on how much light there is and how close it is to the sensitive peak of that cone. And that whole milieu of signals comes out of the eye and through the optic nerve to your brain, and your brain says, well, I see this particular balance as green or red or pink or whatever it might be. So what goes in terms of what's reflected from a plant is going to produce a sensation via your 3 color receptors that gives you the sensation of a given color. Some animals only have 2, some have only 1. Actually, we're outdone by birds and butterflies, some species of which have 4 different cones, and they have a very correspondingly more delicate sense of color than we do. There's kind of a basic fact about light, and that includes everything from X-rays to radio waves. The more wiggles it has in a given space, the more energetic it is. One way to think about it is the energy per wiggle is constant. So over here, ultraviolet light with close together frequent wiggles has a lot of energy, and as a result it does things like wreck chemical bonds. It's not a very good thing to perceive with your eye because it destroys the very molecules that would sense it. Over here in the infrared, it doesn't have quite enough energy to do that. It just kind of gets things warm. And so ideal for seeing the world is visible light, which interacts with molecules that we're going to talk about and produces our ability to sense color. So here's an illustration similar to what we're going to use again and again tonight as well. If you illuminate something with white light, like the light that I'm seeing here that illuminates the scene up here for the camera, it's a mix of all these colors, from blue to green to yellow to orange to red, in roughly equal proportions. When we see something colored, what has happened is part of the light has been absorbed and withdrawn from what we're going to see. And how we illustrate that is we black it out. So a certain color, in this case paint, due to the pigments in paint, has blocked out everything that's black, leaving just primarily red and a little bit of yellow. And what we perceive that as is this color right here, which is a fairly bright orange-red. If you have a little different pigment and you allow a bit more of the green and yellow to come through, you still black out all this stuff. Our perception from this band of colors is to see something that's kind of amber. And if we allow a little bit more of the spectrum to come through, including a fair amount of green and even a little bit of red, what we see is yellow, which is kind of in the middle. So if I had a yellow laser and I could illuminate your eye with that yellow laser, you'd see something like this from that single color. But the same balance of outputs from the 3 cone types is achieved by having this band of color. So pigments color things by removing some of the light and leaving the rest of it for you to see. So what about white objects? What kind of pigments do they have? Well, white is the color we perceive if we're getting all the wavelengths back. So nothing's happening to any of the wavelengths. They're not being absorbed. They don't have any pigments. And here's some examples of flowers that are primarily white. So when you illuminate them with sunlight or another white light source, what you see is white stuff, white flowers. Here are a couple of lupins. The one on the left is a normal lupin with colors to attract insects, the dark blue, and colors to tell insects don't bother. This purple color reflects the fact that that particular blossom has been visited and pollinated. Where up here, these are still eligible for pollination and trying to attract the insects that will do that. This particular lupin did not have genes expressed that made the pigments. Lacking the pigments, it looks white. So that's the pigments provide color by removing some of the incident light in a way that selects against the colors they absorb and leaves the colors remaining to be reflected to your eye. So we already mentioned pigments, and that's really the substances that interact with color in a way that allows us to perceive color. And there are dozens and dozens and dozens of plant pigments. But fortunately, rather than just having this confusing array of many pigments, We can group them into 4, and we're going to look at each of those 4 tonight. Each of those 4 represents a major metabolic pathway. Carotenoids, orange and named for carrots, come out of what's called the isoprenoid pathway. Anthocyanins belong to a group called the flavonoids, and they come out of the flavonoid pathway, or an alternate name is phenylpropanoid pathway. Betalains, which are common to one particular order of plants, come out of the betalain pathway, and chlorophyll out of the porphyrin pathway. So even though there are variations on this, these 4 particular plant pigments cover much of what we see, even though there are variants within each particular group. So now comes a little chemistry, but it's fun. I have to give you one of the quotes I really like, because I like chemistry a lot. Most people hate it. I hope by the end of tonight you like it a little better. Linus Pauling, who was one of the greatest chemists in history, famously said one time at a speech he gave at Berkeley, he said, "Chemistry is wonderful. I feel sorry for people who don't know anything about chemistry. They're missing an important source of happiness." So I want everybody to get a little bit of that source of happiness tonight. What we've illustrated here is a very simple molecule, a carbon atom joined together, 4 hydrogens stuck on, and the gray cylinder in the middle, even though it's a piece of plastic, represents the bond that holds those 2 carbons together. Otherwise, they'd like to fly apart. That bond consists of a pair of electrons that's perfectly happy to spend most of their time right there. They've taken up residence right there. And I'll make the analogy that they occupy a particular residence, and they attract with their negative charge a little bit this positive nucleus and this one, and they hold the whole thing together. Now, in addition to that, if there are other electrons available, they can take up residence in these more distributed zones. And they're called pi bonds, P like the Greek letter, pi. And they can, if you extend this network, can actually spread out and form what I would call analogous to a neighborhood. So if these 2 electrons right here have to stay home and hold the 2 carbon atoms together, electrons that occupy these pi bonds can spread out throughout this extended neighborhood, and they get to roam all around the place. Well, that gives them the wonderful property of being able to absorb light in ways that give the pigments their properties. If this bond absorbs energy, it breaks, and the whole thing comes apart. These bonds can absorb energy, and the whole thing doesn't come apart. But they do absorb light of a particular color. So light shines on these pi bond system. It gets absorbed. What happens to it? It gets dissipated in other forms. It comes out as lower wavelength light. It moves electrical charges around. It's dissipated as heat. So something disappears. So when we shine white light on these pi bonds, they're pretty selective. They will absorb some of the colors and leave the other colors alone. And that's the basis for all of the pigments that you see in plants. So, just to kind of sum up the whole point here, pigments absorb light. And therefore, the light that you see comes to you as colored because it's not the broad spectrum of things that adds up to white in your perception. And pigments absorb light because they have these pi bond systems in them. So, now we're going to look at that was a simple molecule. Let's look at a little bit more realistic pigment molecule. And here's kind of the stick figure that chemists enjoy using. And these are all carbon atoms, a nitrogen, 2 hydrogens, a couple of oxygens. And to make it a bit more shorthand, in common in chemistry, to simply leave off all the C's. In organic chemistry, there are so many of them, people get tired of writing them. So everywhere a line ends or bends, you can imagine is a carbon. That's what this stick figure means. So let's boil it down to the basic framework that holds it together. These are the bonds, like those 2 electrons that stay home in between the atoms and hold them together. A 6-membered ring, and a nitrogen sticking off this side, and a couple of oxygens off this side, and a few hydrogens stuck on for good measure. That's the basic structure of this molecule. But it also has these extra electrons in the pi bond system, represented by alternating dashed lines. This represents a pair of electrons, but keep in mind they don't stay here. They flow all around the place. as we can illustrate with this yellow diffuse zone. This represents the neighborhood, and each pair of electrons can wander anywhere in that neighborhood, and they're very good at soaking up light. That particular part of the molecule is called the chromophore, which means color carrier. And so this is the portion that interacts with light. This particular molecule interacts with ultraviolet light, and you probably know it. It's PABA, which is an ingredient in many sunscreens because it absorbs ultraviolet light. Now, for a given structure, the bigger you make it, the more you will absorb lower frequency light. So here's a good rule of thumb to help understand what we're going to talk about. The bigger the electron neighborhood, the bigger this chromophore, the bigger the wavelengths it will absorb, the longer. Therefore, it's going to take up a little more red and leave a little more blue, and objects will appear to you more blue and less red. So after the pigment soaks up what it's going to get, what you get is what's left over. That's the color you'll see. So let's look at a representative of each of our four main types of pigments. First, beta-carotene, which is a carotenoid, and it looks like this. And notice all these little pi bond representatives here. This is a much more extended pi bond system than the one we just looked at. So, would you expect it to absorb longer or shorter wavelengths? Longer, right. And one analogy you can keep in mind that actually I think makes good sense is larger musical instruments play lower notes. They would also, in the reverse, absorb lower notes. The larger, say, a stringed instrument is, the more it operates in the bass end. And the same is true of the chromophores. The larger they are, the more they operate at the bass end, the longer wavelengths they absorb, and the bluer they leave the object. This is the absorption spectrum of carotene, and it's really a fairly simple graph, similar to what you already looked at. Across the bottom is the spectrum from blue to red, 400 to 700 nanometers, and this line represents how much is absorbed. So it absorbs best right in the blue. It doesn't absorb worth a hoot out here in the yellow and the red end. So it's kind of hard to picture what's left. So the way we'll illustrate this is we'll look at not what it absorbed in the form of this graph, but we're going to turn this into a picture with what it absorbed missing. So what's black is gone from the light that you'll perceive when you look at that particular object that's colored by carotene. So what's left? Not very much blue, a lot of green and yellow and red. So what kind of colors do we get out of things that are colored with carotenoids? These kinds of colors. Certainly yellows. If you adjust the carotene's environment and its composition a little bit, it can become quite orange. Here's our wonderful Frumantia flower, bright yellow. All of this is carotenoids, these yellow maple leaves, these big leaf maple leaves. Even the tomatoes, a carotenoid called lycopene, absorbs a little more of the blue and a little less of the red. So all of these are examples of coloration by carotene. Now, all I've shown you here is just what carotene in solution in the chemist lab or the physicist lab would absorb. In real life, it's embedded in the tissue of a plant, and the absorption is broader than what I've shown you here. But it gives you an idea of why that dominates the color absorption. So, the next major group, the anthocyanins, a flavonoid. They have three rings like this joined together and lots of pi bonds, and they're linked together in a system, and the chromophore in this one looks about like that. It's pretty good sized, and it absorbs pretty well in the visible part of the spectrum. If this is too small, it absorbs like PABA in your sunscreen, ultraviolet, but not visible. There's the absorption curve. It peaks in the green, not very much in the red, not so much in the blue. And if we show that as a spectrum with the absorption blacked out, it looks something like this. So uniform illumination with white light would give it all these colors. The anthocyanin would absorb a lot of the green, leaving blues and purples, reds and yellows. here's an example of the kinds of coloration produced by anthocyanins. It's typically the blues and reds and purples that you see in flowers and plants. From beautiful dark blue, this is indigo bush from out in the desert. Another blue flower, this is red maple, dogwood, California grape, some berries. What are those berries? Those are toyon berries. Yeah, that's it. This beautiful pink from the owl's clover. So you can see that's a little different than what you get from the carotenoids. Now this is a really interesting one to me. It's called betanin. It is a beta-lane. This is one particular one. It occurs exclusively in the order Caryophyllales. And that includes cacti, 4 o'clocks, bougainvillea, beets, hence the name, and a couple of others that don't come to mind right now. Chenopodes. And this is the chromophore. It's a totally different kind of pigment. It's a nitrogen-containing pigment, totally different in structure from the ones you have seen, and unique to this particular plant order. They don't produce anthocyanins. Their version of a red pigment is this. Here's its absorption. Well over here in the yellow and the green. So in the 3 that we have considered, we've seen the absorption get longer and longer and longer in wavelength, and that accounts for the shift in the colors that it produces when it dominates a particular plant's spectrum. Once again, we look at all the colors. We take out the part that's absorbed by the beta-lanes, leaving a lot of blue and purple, a lot of red, and these are the colors that it produces in plants. There's the classic beets, deep purple red. This is pinks, that's another family within the Caryophyllales. Nice red. This is Opuntia, cactus. And 4 o'clocks, this is spiny hopsedge, one of the chenopodes out in the desert. More bougainvillea. So, it has some beautiful colors, but only within that particular set of families. And we can't leave out good old chlorophyll. Now, when we talk about plant colors, we tend to think, flowers and reds and yellows and blues and things like that. But what's the dominant plant color? Green. And chlorophyll does it. It's a porphyrin, very different. It's a big flat ring like this. Lots of the pi bonds all knitted together in the system. That makes a chromophore that spreads out like that. And in the middle is a magnesium atom, which affects the nature of the color. In a way, you can think of our large musical instrument being fine-tuned by what's around it, and so it shifts the absorption a little bit over what it would otherwise be. Chlorophyll absorbs very well in 2 parts of the spectrum, the blue end and the red end. And somewhat interesting is only the photons at this energy level are useful for photosynthesis. All the energy absorbed here is dumped as heat until it gets down here, and then that's what's channeled to the photocenter. So I think it's a nice example of the fact that evolution is what works. It isn't what's perfect. And so here you have the dominant pigment on the planet that makes all the rest of the life possible, absorbing strongly in the blue and doing nothing with the extra energy. It degrades it to this energy level before it's used. But anyway, let's look at the absorption spectrum. It takes out a lot of the blue and purple, and it takes out a lot of the red. Now, again, these are spectra that are done in the laboratory in a given solvent. In the plant environment, these peaks are much broader. It actually takes out quite a bit more of the red than those skinny peaks would suggest. And I know you're going to guess the colors we get out of chlorophyll. It leaves a lot of green, and there you have it. It's present in algae, mosses, seaweed, angiosperms, simpler plants, stems, leaves, sometimes the fruit, and it is the pigment that absorbs the energy of sunlight and turns it into chemical energy in the form of, ultimately, sugars that lie at the base of all the food chains except some of the chemosynthetic ones out in the middle of the ocean. And I think greens are just as cool as the other colors, actually. They really vary in their shade and their intensity because of the rest of the leaf to a large extent. But especially in the spring, we see this gorgeous palette of green colors that vary from sort of bright new green to the older green of, say, conifers, where the leaves, the needles are not brand new. So, we talked about the basic absorption and therefore the basic color imparted by the four major pigments. Now, life isn't simple, and there are fine-tuning things that go on that will shift and change the colors, and that helps account for the broad range of colors that we do see. So let's take a look at the several things that are very important in changing color. If you change the size of that pi bond system, change the size of the neighborhood where these electrons can roam around, you'll change the absorption. And here's a good example in the carotenoids. First, we look at one called phytoene. Now that has a lot of these bonds, but look, there's a gap. There's a gap. So the neighborhood that constitutes the chromophore is really very short. Small chromophores absorb small wavelengths, and this absorbs here in the ultraviolet. It doesn't even get much into the visible. But in the case of violaxanthin, which is a carotenoid, the xanthins are carotenoids that have added an oxygen someplace. It absorbs much more strongly in the blue. The chromophore is much bigger. Our old friend beta-carotene absorbs a little bit longer wavelengths, more toward the green. And lycopene, which is in tomatoes, absorbs right here in the blue-green part of the spectrum and leaves a whole lot of red. Where beta-carotene would absorb a little less of the yellow, and so the yellow would combine with the red and give you a sensation more like orange, which is the color that you see. So the whole point of this particular slide is, as adjustments take place in the extent of that pi bond system, then you change by fine-tuning the absorption, and therefore you fine-tune the color that that pigment will produce. We can use the anthocyanin in this example of how pH, the acidity or basicity of something, changes it. You gotta look quick here, right up there. Let's suppose we put this in a very basic environment. Basic environments like to hog up hydrogens, and so they're going to pull a hydrogen off of here, and it's going to extend. I'll go back. This is not part of the pi bond system right now, but when we take that off and make that a double bond and all these flip around, then this becomes part of it, and it's extended the neighborhood a little bit. I'll show you in a bit more detail. This part of the molecule had an oxygen and a hydrogen on it. If we put it in a basic environment, it's going to take the hydrogen off. The extra electron pair is going to flip into place here and extend the pi bond system. Pi bond system gets bigger. The wavelengths it absorbs gets bigger. The object gets bluer. And you have a nice illustration of that right down here, using that very compound in solution. Here's pH 5.5. That's more basic. This is more acidic. And as you put it, go from an acid environment to a basic environment, and you start pulling all those H's off, it begins to absorb longer and longer wavelengths, leaving more and more of the purple. And something that's kind of fun you can do yourself, and it was used a lot in the past. Plant pigments have been used as a pH indicator many, many times. And you can go out and pick some blackberries or some other dark, juicy source of anthocyanins, and just dissolve a little of it in water and put some vinegar in it or put some ammonia in it and watch it change color. We once took our grandkids to a couple of places. One happened to be Mono Lake, which has very basic water, and one happened to be Lassen Park, which has very acidic water, and we brought some berry juice along and put it on paper and had a grand time watching it change color so that it reflected the change in the pH. The presence of a metal ion affects the color as well. Here are two almost identical porphyrin rings. This is hemoglobin in your blood. This is chlorophyll in your plants. Very similar in terms of the pi bond system, but this one has magnesium, this one has iron. This one imparts a red color when it's oxygenated. This imparts a green color. And as you know, when the oxygen is taken off, and that changes things too, deoxyhemoglobin is blue. So the color depends not just on that electron system that absorbs the light, but it depends in detail on other things that adjoin it, because that raises and lowers the energy levels and fine-tunes its absorption. So it makes a big difference. And this brings up another example that's kind of fun to talk about. Hydrangeas are famous for changing their color from pale blue to pink when they're put in a less acid environment. And people have often thought that it was a direct result of changing the pH, but it's not. Think about that previous slide. By making it more acid, the solution got redder. But when you make hydrangeas more acid, they get bluer. So that doesn't quite fit with the basic idea of what pH is doing. What's happening is more acid soil will liberate more aluminum, which is absorbed by the plant. Aluminum, the metal ion, helps to make complexes out of the flavonoids that are in there and makes them a bit more blue. So, some of the things that decorate the edges of this molecule also affect it. Here we have 3 anthocyanins. They all have this same basic configuration, only R stands for things that can be changed from one thing to another. And they have names like pelargonidin from pelargonium, cyanidin meaning blue, and delphinidin from delphiniums. And the difference between those 3 is pelargonidin has 1 OH group on here, cyanidin has 2, and delphinidin has 3. And in the process, it shifts it from being a red pigment, meaning it's leaving the red for you to see and hogging up the blue, to being a blue pigment, which means it's taking more of the red and leaving more of the blue for you to see. So here's an interesting detail about how these pigments interact with light. This is a cross-section of a leaf. It's ivy. That's the top surface of it. So this row of cells right here would be the epidermis. This is a palisade layer, kind of short palisade cells, but palisade nonetheless. And the more spongy layer down here. And the dark objects are chloroplasts, which is where chlorophyll resides. Now, if light, we say, well, light's reflected back to my eye. And the first thing you think is, well, it's reflected from the surface. But if it was reflected from the surface, what you'd see is just shining. You wouldn't be seeing any absorption by the pigments because the light didn't reach the pigments. So what really happens in most of what you see is that light, here we've illustrated it as white light, and then we've broken it into a very simple spectrum of blue, green, and red, comes down, goes through some of the chloroplasts, and when it comes out the other side, the green is still there because chlorophyll doesn't absorb green, but it's red and the blue are much diminished, goes through another couple of chloroplasts, the red and the blue are essentially gone, and it bounces off a space down here where an air space adjoins a cell. That's a great place to get a little internal reflection from. And out it comes, goes through a few more chloroplasts, which actually do darken it some. They do absorb some of the green. Hits another little reflection point here, and out it comes. So what went in was white light, and if that just bounced off the surface, what you'd see is white light. But by the time it gets to ricocheting around in here and having its red and blue absorbed and its overall intensity diminished, what comes out the other side is a fairly dark green. So what do you see when you look at that leaf? You see something fairly dark green. Here is, now up to now we have looked at these spectra of how a single pigment will absorb light. In the plant itself, things are a little more complicated, and so what we see here is the absorption spectra of a leaf. So, for blue to red again, the higher you go here, the more light the pigment is taken in and stealing and not letting you see. So we're going to color that black. That's what gets taken out. Notice it's not a peak anymore. It's kind of a broad absorption with an edge on it. And a little bit more green left over for you to see, green and yellow. And if you looked at a leaf like that, it would look something like this. The shiny parts are where the angle of the leaf is such that you're getting a lot of reflection from the surface, and you're not seeing deeply into the leaf, and you're not seeing the green. But most things are not near as bright as the sun when you look at them. They're greenish, and they're much darker, and the reason is that light has taken quite a path through the leaf before it turns around and comes back in your direction. A couple more spectra of whole leaves to show you how that compares with just the pigment. You remember this pigment right here? That was the first one we looked at, beta-carotene. Absorbs strongly in the blue, but if you put it in a leaf, there's a lot of other stuff in that leaf that are absorbing light as well. And so what it does is it just sort of influences where this begins to drop down and you get a lot of light left over. This is the part that is lost to all the pigments in the leaf. This is what remains, and when this comes out of the leaf, what your eye perceives is yellow. That's a fall color of a leaf. Actually, this is the spectrum of this leaf. It's dominated by carotenoids, but that's not the only thing in there. So when you get a lot of yellow and red and some green, what you see is yellow. Another one, in this case, a leaf colored by anthocyanins, fall leaf. This is red oak. Here's the absorption of just the pigment. Here's the absorption of the leaf. We color black the part that got absorbed and you don't get to see, and what's left over is this. It looks almost like this, but there's less green and yes yellow compared to the amount of red, and what we perceive is a nice red fall leaf. In this case, red oak. So, interesting question is, where are these pigments? That has a lot to do with how we try and explain what they do. Here's a plant cell. Nice schematic of one. This is the cell wall of the plant, and it's full of all kinds of wonderful stuff inside. Organelles like this, and this, and this. These are mitochondria. This is a chloroplast. All kinds of membranes weaving around in here. And so we can ask, where's the chlorophyll? Well, it's not everywhere in that cell. It's in here. And these are the chloroplasts. This one, this one, this one. They are absolutely full of internal membranes, and that's where most of the chlorophyll is. In photocenters, and what they call light harvesting complexes, the little antennas that help it absorb light. And it's kind of interesting to ask, well, how are they arranged in there? And this is just part of the detail, but it gives the idea. This is a schematic of one unit of basically an antenna. Chlorophyll is not terribly efficient at absorbing light. It takes several hundred chlorophyll molecules to feed one, what's called a reaction center, which then goes on into the metabolic pathway and begins to make sugar. And each antenna consists of 3 parts, and this is one part. And all these little squarish things here are the porphyrin ring of a chlorophyll molecule, all arranged just so. These spirals represent the protein that surrounds it. And can you see those things, yellow and long? What do they remind you of? The molecule that we looked at. The carotenoids. So carotenoids are an integral part of the system that includes chlorophyll as antennas and as reaction centers, so they're always present there. When you see the green of chlorophyll, you're also seeing the effect of a certain amount of carotenoids as well. When you see a spectrum of chlorophyll published somewhere, they squashed it and took it out of a leaf and put it in a solvent in a lab and put it in a little glass container and measured the spectrum. But in real life, carotenoids are always intimately associated with the chlorophyll. And this arrangement is exquisitely precise. This is not just some hodgepodge. One of the really wonderful things about this whole process is just how well and how efficiently the light that's absorbed is translated into energy that can transfer all the way to the reaction center and ultimately separate some electrical charges and make this whole thing work. It's extremely efficient, and it's efficient because these things are placed as carefully as if they were precisely machined and put in there. It's not just thrown together in some solution or some soup of chlorophyll and other things. So if you're trying to explain how a given molecule such as this affects the absorption of light, it has to be in the context of keeping in mind this really exquisitely precise and efficient structure. And in fact, these carotenoids do play a role. They help absorb some of the light that chlorophyll can't see, and they dump the energy to chlorophyll. And they also serve as a protectant in that any photosystem can be overdriven. It's dangerous business turning light into other things. It's high enough energy to change the chemistry within these molecules. And that means you generate a lot of things like free radicals that can cause problems. And so if you get too much light and you can't process it out the other end fast enough, it does a lot of damage. And part of the protection against that are these carotenoids that are embedded within the system. Well, where are the carotenoids? Well, I already told you where some of them are, right there. But they're an integral part of the photosystems and the antennas. But what about when a leaf turns nice and yellow, or a tomato turns nice and orange or red? Those carotenoids are in the chromoplasts. These are chloroplasts because they're green. These are chromoplasts because they're colored. And the carotenoids are membrane bound for the most part. They're not very soluble. And so when a cell becomes highly colored, like in a yellow part of a flower or a yellow part of a fall leaf, much of it is contained within these little things called chromoplasts. Well, what about the anthocyanins and the butylanes, the reddish pigments? Well, they don't go there at all. They end up in the vacuoles, which are these sort of large spaces within the cell that contain a lot of water, because these are water soluble. They don't want to go in the membranes. Those are sort of places for oily molecules. These are water soluble. And so sometimes people make a quick effort to explain why, for instance, red color in fall leaves has something to do with photosynthesis. Well, it doesn't, because photosynthesis is going on over here, not over here. It does help protect the photosynthetic apparatus in certain cases simply by shielding them from so much light, but it can't participate in the actual production of sugars and part of the photosynthetic antenna system because it's not there. So let's look at some changes where, let me go back one step. These are plastids. They're descended from an ancient bacterium that got stuck inside of a cell somewhere probably a couple of billion years ago and now perform special jobs. Mitochondria are a different kind. But these and the chromoplasts are very closely related, and it turns out they can morph one into the other, which is really kind of cool. And here's an example of that very thing. Here's a tomato, started out nice and green. Why is it green? Because having more chlorophyll going to work doing photosynthesis in the entire skin of that tomato helps produce the sugars and so on that the plant needs to grow the fruit. But then it ripens, and it decides it's not in the business of doing photosynthesis anymore. It would rather be eaten, so it wants to look really attractive, and it starts getting not so green and becoming much more yellowish or reddish. Similarly for the lemon, it was all green, and then it starts in the area that's most light struck. It starts to turn yellow. And here's a pepper that was green, and it's all but lost all its green, and it's now becoming red. All of these are happening because chloroplasts are becoming chromoplasts. And here's one caught in the act. This is the coolest picture. The plant cell in this case is really big. But this is just the chloroplast. This is an electron microscope picture, bounded by its little membrane. These membranes are what's left of a chloroplast. Chloroplasts are just full of layers and layers and layers of membrane. That's where the photosynthetic machinery is placed. And so what it was being a chloroplast, this is the machinery that did it. But these are dissipating, but you can see its chloroplast heritage because of these membranes. But now it's becoming a chromoplast, and all of this is carotenoid. And before it's over, all these membranes will be gone, and this chromoplast will be largely full of carotenoids. So it's changed its job from production of energy to the production of color to make the tomato look good, so we eat the tomato and distribute its seeds around. So when you look at a piece of ripening fruit, it's easy to say, oh yeah, it was green and now it's kind of ripening and it's getting yellow. But think about what's happening. It's amazing. The seeds are becoming mature enough that this fruit needs to be eaten so the seeds can be processed through somebody's gut and distributed some nice place. And that somehow signals the skin of the fruit. And cells in the skin of the fruit signal the cell's genome, the cell's DNA, to turn this chromoplast into a this chloroplast into a chromoplast. And when that happens, the DNA within the chloroplast, now becoming chromoplast, goes to work actually producing the carotenoids. So what you're seeing is not just, oh, the tomato's getting yellow. You're seeing an astonishing, complex series of events, signals that go from seeds to skin, from the nuclear genome of a plant cell to the plastid genome of the plant cell, and a chloroplast morphs into a chromoplast. And it can go the other way. When a carrot, for example, sticks above the ground and it ought to be nice and orange and it turns green because the plant says, well, I'm out here, I might as well do something, and so I'm in the light, I guess I'll become a chloroplast. So there's a whole lot more to think about than just, oh, it changed color. So let's talk about that's a lot about the general processes, the chemistry and physics of how color is produced and some of the molecules that are responsible for it, and some of the places it takes place, and some of the things that can fine-tune the spectrum and therefore fine-tune the color. And now we'll just look at organs of the plants that are colored and ask ourselves a little bit, why is that? Well, here's a whole set of beautiful flowers. And if I'd had more time and you had more time, we could put pictures up here till we've been here a long time showing the beautiful colors of flowers. And they can be just gorgeous. This is one of my favorites. It's desert Mariposa tulip. And Mariposa tulips tend to be sort of a pale, subtly colored thing. But out in the desert, they are just this blazing, deep red orange. Here's alpine gold, which is a gorgeous alpine plant with big, beautiful yellow flowers. Cactus. There's just no end to these things. Well, how come they're so pretty? And I guess you know the answer already. To attract pollinators. Here's one, he's been attracted. Here's another one having a great time. There's a little tiny one. Here's another one. So they put on a heck of a show to attract beetles, butterflies, flies, wasps, bees, all kinds of things like that in order to transfer pollen and be pollinated. So that's a pretty simple story. If someone knows about other colors, and we have a chance to talk about some of this after we're done here, let me know. Leaves have lots of things going on due to plant pigments, and they go through a number of stages. They develop, they operate as mature leaves, and then they senesce. And they aren't just colored with one pigment, they're actually colored with a number of them, depending on where they are in their life stage and ecologically what demands are being placed on them. So we'll talk about some of those things. Here's a collection of leaves. They're all green, pretty green. That's not very green. That's not very green. But the chlorophyll down inside is green. Why they look somewhat like this or somewhat like this is a surface effect. Here's a good example. This is called desert holly. It's an atriplex. It doesn't even look like it has green leaves at all. But if you tear one of these in half, you'll see this beautiful dark green little stripe inside that is the layer of cells that contains the chlorophyll. But these happen to have a surface that reflects a lot of the light. This is artemisia, and so does it. This is another atriplex, and so does it. If the surface reflects a lot of light, the light rays are not going to get deep in that leaf and get colored green by making a passage through the pigments. And so what you're seeing here is a lot of light coming back from the very surface, not penetrating, and that's the whole point of the plant being so white. It's trying not to absorb too much light, because this grows in astonishing places, out in black rocks at low elevation in the Mojave Desert when there's nothing else around. You just see these white patches and you think, what in the world is that? It's this. And it's alive. It's amazing in what it does, but it can't overheat. So it sends most of the light back right away, lets a little bit through to the chlorophyll, and that's all it takes. So let's look a little bit at where the chlorophyll is in a leaf. Here's the cross-section of a leaf in the microscope. There's not any up here in the epidermal layers or down here. There is a little bit in the guard cells of the stomata, but for the most part, this is not the chlorophyll-bearing layer. It's in these long cells called palisade cells here. Sometimes there's one row, sometimes two, sometimes three. And in the so-called spongy layer down here, which the cells are more open, they have lots of air passages to let carbon dioxide in and water out. And so in a leaf that is functioning, that's doing its thing. It's a nice mature leaf. This is pretty much what it has. Chlorophyll here, and it doesn't need any other plant pigments except the carotenoids that are part of the photosystems. But sometimes plants have both chlorophyll and anthocyanins at the same time in a mature, functioning, healthy leaf. And it looks something like this. Here's predominantly green chlorophyll, but up here in the palisade layer is a lot of anthocyanin, and you can see that by the redness. And so you see examples of this all the time in the form of things that look purple when they're not in their fall colors at all. This is really common for some horticultural varieties of things like plum trees, and it's because both pigments are present. If you go tear that leaf open and look carefully with your hand lens, you'll find the green layer and the purple layer. Usually the purple is either on top or both the top and the bottom. Another example of where both anthocyanin and chlorophyll is present is in newly emerging leaves. Now, a brand new leaf, just making its chlorophyll and its photosystems, is getting a lot of light, but it's not ready to process all that light. The light part can overdrive the system if the excitations, the energy that's being dumped into the photosystem isn't used up in the metabolism that leads to sugars. And that metabolism depends on having everything in place to make it work. It depends on high enough temperatures to make it work. And so here's Photinia. It's not a native plant, not native here anyway. Very, very common. The leaves that come out initially look red. And they look red because even though there's chlorophyll inside them, and they're developing and they're trying to get up to speed, they need some protection from excess light. And one of the things that anthocyanins do, probably the main thing that they do, as far as people can determine, is they are a light screen. They help diminish the amount of light that's impinging on the chlorophyll. Their absorptions overlap enough that they can be helpful that way. And so these are just as red as can be, but they lose that redness. Here in the microscope is an example of another leaf. This is cocoa, a new leaf that has both anthocyanin and chlorophyll. And if you look closely, you see a greenish tinge all around here. This is the cells of the mesophyll. And the redness is in here. This is the vascular tissue. So this happens to be where the anthocyanins are produced, but they help screen light throughout the whole leaf. So here you're seeing anthocyanin as an accessory to help protect the newly developing leaves. But then trees eventually, after they get mature, and leaves I should say, in deciduous trees, they senesce and then they're lost. And we often see fall colors begin on the upper outer parts of trees, and this is something I'd invite you to spend a little time noticing when fall finally comes to this part of the country of ours, is not all parts of the tree become colored at the same time. They tend to be the outermost and most light exposed parts, or sometimes particular branches. This is the Chinese pistache tree, which is really nice in that it has beautiful fall colors even in very temperate climates. It doesn't take the cold of the Northeast or Canada to make it pretty. Another example, this is a flowering pear, but again the most light exposed parts start to turn first. This is sugar maple in the upper lakes country of the United States. And some of the branches, obviously, the genetic nature of those branches is such that they get the signal a little bit earlier of short days and cold temperatures, and they start to turn first. And they turn on their outermost parts. These are becoming red because anthocyanin is developing in the leaf. These are yellowish because chlorophyll is leaving, but the anthocyanins have not fully developed. And these are still kind of green. So it's progressing first up here and gradually down, and overall the same thing, the whole tree. Here's beginning to change color, beginning to change color. This is much further along. The darkest red, where it's mostly anthocyanins, is out here at the end, and a combination of anthocyanins and carotenoids further down. So in close-up view, what's happening, here's a cross-section of a leaf, a diagrammatic one. That's the top of the leaf, that's the epidermis, doesn't have any pigments in it. The palisade layer, where a lot of the chlorophyll is, and the spongy layer down here, and this is the vascular tissue. So first you have a leaf full of chlorophyll. That's before it senesces. And it's green because it's mostly chlorophyll and a little bit of carotenoids. Now the chlorophyll begins to break down as the leaf senesces and gets ready to fall off the tree. So next, what you might see is it looks more yellow. And it's often said the chlorophyll is disappearing and unmasking the carotenoids. That's one way to think about it. I tend to think about it as if you have two pigments, you're going to get a different color than if you have one pigment. And if you subtract chlorophyll to green pigment, you're going to see more of the yellow. Likewise, if you subtracted the yellow, the chlorophyll would look a little bit different to you, but that's not the direction it goes. And when the chlorophyll is about half gone, you start to see the anthocyanins build up, and that begins to dominate the color of the leaf and make it reddish. So why is this happening? How come anthocyanins are developing anyway? This leaf's about to fall off the tree. And so why invest in this? It takes energy, it takes materials to actually do it, and yet many, many trees do. And lots of research has been put into that, and I have to be honest and say, even though there are some pretty clear conclusions about it, the whole story really hasn't been told. But an important part of it seems to be that these are protective. Well, what are they protecting? This leaf's about to fall off. What they're protecting is this organized retreat that the leaf is performing. The leaf contains a huge amount of protein. The most dominant protein on the earth is the protein that helps run photosynthesis. And so the tree has invested a lot. Proteins contain nitrogen. Nitrogen is a very precious element to plants and the thing that's often in short supply. It makes everything else out of carbon dioxide and water, so there's plenty of that around. They're getting to be more and more carbon dioxide, actually. So there's an investment here that the tree would like to recover, and there is a very organized breakdown and scavenging and recovery of much of the material that makes up the chlorophyll and related proteins. And so that isn't done willy-nilly. That's done in a very specific way with certain enzymes doing what they have to do, and the chlorophyll continuing to perform some of its duties, and as the system is taken apart and is still receiving the full light, it just starts ruining things. And so if the tree is to make efficient use of this recovery process, it needs a little cover from all that excess light. At this time of year, things are getting cold. As the temperatures go down, it really slows down the metabolism that leads to producing things, but it doesn't slow down the light absorption, and so things get out of balance. And so anthocyanins have a role in protecting the leaf's metabolic mechanisms as it extracts those nutrients and gets ready to fall off. The carotenoids, it doesn't bother recovering much of those. Those are just made out of carbon and hydrogen, a little bit of oxygen, so it doesn't take those. They stay in the leaf. Anthocyanins infuse the leaf, and finally the thing falls on the ground. Now there are a couple of other ideas about anthocyanins. One of them, interestingly enough, is that maybe it helps prevent insect eating the leaf. And some experiments have been done in which there's a slight tendency for aphids to lay fewer eggs on leaves that are really red. And the interpretation is, well, this really red leaf says this plant is really robust, really good at doing what it does, and so it's got all this anthocyanin out there helping protect things, and you better not mess with it. And so they go put their eggs on less red leaves. I think the evidence for that is probably a little sketchy, and about the time you convince yourself that's the reason, then you have to explain why the plant next to it that doesn't make red leaves at all is getting along fine. In fact, that's part of the question about, well, if anthocyanin is doing this, how come we have leaves that are just yellow? And I don't know the answer. I don't know that an answer's been put forward. Maybe it does other things better that compensate. So now let's look at some of those senescing leaves, those that are losing or have lost their chlorophyll. We looked at a nice developing and a mature leaf before, nice and green. Well, here is one that's pretty much lost the chlorophyll. It's been broken down, it's been retrieved, things that are useful in it have been recovered by the plant. Mostly carotenoids down here, but a lot of anthocyanins in the palisade layer. So this leaf would look pretty reddish to you if you saw it on a tree in the fall colors. Here's an example of one that doesn't have any anthocyanins, just the yellow carotenoids left in the leaf. And that would look yellow to you, and some leaves are yellow, like beech leaves and aspen leaves. So here's an interesting way of looking at the variation in color that comes about as the balance of the pigments changes. This is off a flowering pear. This is a leaf cut in half, lower surface here, upper surface here, that was just beginning to change color. And the first color it changed to was purple. And remember, purple is really commonly produced by anthocyanins in the presence of chlorophyll still remaining. And if you look at the bottom of the leaf, the dominant pigment is chlorophyll and it looks green. If you look at the top of the leaf, it's purple because you're seeing a lot of anthocyanins overlying on the top surface, in the upper part of it, the chlorophylls. A little while later, that same leaf will look like this, where on top you're seeing red, not because anthocyanins have changed, but because the chlorophyll has left. So if you have two pigments and you get purple, and you take away the green one, then you're looking more at red. And so the anthocyanins dominate the appearance of this leaf, but you flip the leaf over, and on the bottom, it's dominated by carotenoids. You can see a little red peeking through, but that's mostly yellowish. And that makes sense in terms of the role of anthocyanins being protective against excess light as the leaf senesces. They're mostly on the top. That's the side that gets most of the light. And they've done experiments where they shine light from the bottom and the top, and have shown that light from the bottom isn't screened near as well, and there is an effect on how well the plant recovers nutrients from that senescing leaf. And here they are all put together in 4 different colors. So this is upper sides with chlorophyll and anthocyanins, no chlorophyll. Lower, chlorophyll dominates, makes it look green, but when it goes away, you're left with all those carotenoids, but not much anthocyanin down there, because the bottom of this particular species of leaf doesn't develop very much. Something you can notice for yourself really readily, and this is one of those things that's kind of fun to look at, is that anthocyanin synthesis requires light. There's plenty of anthocyanins here, this is dogwood, and none here. Why? Because another leaf was lying close against the top of this one and shaded it. Right here, this little berry, this little miniature pear, shaded this part of the leaf and left it devoid of anthocyanins. And here's dogwood again. This was back further in the plant and a little bit more shaded, and this is the leaves that hung out in full sun. Same thing here. This is green because chlorophyll's still left in it, and yet this was shaded and it's not developing anthocyanins. So one of the triggers, the signals the plant uses to say, hey, I better make some anthocyanins, is high light levels. As you enter the fall, you've still got plenty of light, temperature slowing things down, you start to get things out of balance, light drives the system too hard, metabolism can't use all that energy because it's cool, and the leaves are beginning to senesce, and so the plant says, hey, wait a minute, this is the time I need to develop some of that light screen, some of that anthocyanin. But if you deprive it of the light signal, it doesn't get it, and it doesn't develop it. It needs sugar. It has to have the energy and the materials to actually synthesize anthocyanins. So ideal is cold nights. It gives it a cold temperature signal. That tells the plant, hey, winter's coming, you got to do something about it. Bright days, plenty of light that tells the plant, you know, things are getting out of balance and you better do something. You're going to wreck your system before you've dismantled it. And warmish enough to allow the metabolism of sugars and the metabolism that produces anthocyanins. So that's why you often see quite varying fall color shows, this interplay between what was the temperature regime leading up to it, how much water has the plant had that year, how bright are the days, do we have a lot of cloudy days, do we have a lot of sunny days. All those things play into this complex set of signals that tell the plant it's time for you to make this red stuff because that's a good idea, because you'll be better off next year because you've recovered more of the usable materials from your leaves. And that's just a nice picture that shows the whole variation of color in a dogwood, from bright red, sun-exposed leaves, fully developed anthocyanins, to green leaves that haven't quite started yet, to yellow leaves that have lost their chlorophyll, some just tinged with a little bit of anthocyanins, but the carotenoids showing through. And it's really wonderful that you can take these three pigments and you can vary their concentrations a little this way or that and get this beautiful mosaic of plant colors just in the leaves as they're starting to senesce and end their season. Now, they don't change color all at once, and that can be kind of interesting and revealing also. This is red oak, and the areas between the major veins have changed color, have a lot of anthocyanin, and rather quickly they become kind of brownish, which means that a lot of tannins and a lot of oxidized compounds are coming into play, and they tend to make it just dark and brown. But along the veins is still green. These are the areas from which not all the good stuff has been taken and where not all the chlorophyll has been recovered and scavenged. Same thing here in red bud. It doesn't have much anthocyanin, a little bit around the edges, but most of this is carotenoids. But once again, the green remains along these conduits through which the recovered materials are going. Here you got some green and some red, and this is progressing just because this was the most light exposed part. Here's another one, Chinese pistache, that's red on top. That's the most light exposed part. Before it's over, the bottoms will be red too, but the anthocyanin concentration begins to build up in the upper layers first. So if you look around, you can see all kinds of cool patterns in how the leaves go through this process, and you can ask yourself, well, why is the leaf doing this? I wonder why it's still green here, or why it's still yellow on the bottom, or why it's only red on the edges. And yeah, I won't have any answers at all, but it's fun to color. So just some nice pictures of fall colors. California grape, what would you guess was producing the purple? That's anthocyanins with maybe a little chlorophyll left there too. This is sugar maple. Lots of anthocyanins, but lots of carotenoids as well. This is red maple, lots of anthocyanins dominating the coloration. What's that? Ginkgo. Brilliant yellow leaf, which comes partly from carotenoids, but ginkgos do a really cool trick. I have no idea why. They contain a compound that fluoresces. It absorbs ultraviolet light, high energy light, and delivers it back as yellow light. So it actually glows yellow to some degree, and so that enhances the yellow color of ginkgo trees. So there's pigment in here. Maybe that's a protective pigment. Maybe that molecule is absorbing ultraviolet, which can be very damaging. Remember, a lot of wiggles means a lot of energy, and ultraviolet light can rip apart primary chemical bonds and wreck your molecules, which is not healthy. And so it could well be that that's sunscreen, and it happens to get rid of that energy harmlessly enough by emitting it as yellow light. I made that up, but it makes it's entirely possible. Here's a nice ginkgo tree. You look at different trees juxtaposed. Sometimes the combination of colors is really interesting. That's a raywood ash. It's purple, and guess what? Anthocyanins are developing in the leaves. Chlorophyll still hasn't entirely left, and you get that purple color. No anthocyanins here, no chlorophyll left, and an extra glow being added by that UV fluorescent molecule. And this is really nice. This is sugar maple, which really varies a lot in its colors. It can be orange, it can be almost red, it can be brilliant green, it can be sort of a bright green-yellow. And back here, red oak, which, true to its name, tends to get quite red. Lots of anthocyanins here, not so many here. Carotenoids both places. A nice scene, not in California, I have to say. But that's okay. We're talking about plant colors, and they're universal. This is on the Wisconsin River, part of a beautiful scene. Nice waterway, and then the different broadleaf trees turning colors along the bank with green, evergreen trees providing contrast. I never get tired of looking at fall colors. They are so cool. And we don't have to go clear to the East Coast. Here's a western scene with some nice color. Aspen trees of different hues. And just so this didn't seem like this was an East-centric program, here are, well, next slide, a number of scenes from the West. But before we go there, kind of recount, what do the leaf pigments do? A lot of stuff. First and foremost, they are what captures the light energy that runs the rest of life on Earth. That's pretty neat. And they do it with chlorophyll and a little bit of help from carotenoids that are part of that photosystem as well. Leaf pigments also protect those very photosystems from excess light, from being overdriven when they can't deliver their energy down the metabolic pathway and get rid of it. Especially a problem during assembly when the leaves are very new, they're putting those photosystems together, and when they're taking them apart when the leaf is old and ready to fall off the tree. They provide some antioxidant activity. Pigments are good antioxidants because all those pi bonds which are floating around out there and having a great time absorbing light like a bunch of sponges are also very vulnerable to attack by oxidants. And so they sort of take the flak, so to speak, and act to sacrifice themselves to the oxidation by oxidants. And actually, something you might think about, this puzzled me for the longest time, how come chlorine is such a good bleach? Well, because it tears up pi bond systems. It just takes those electrons, which are kind of footloose and fancy-free, it says, come here, takes them away, and that wrecks it. And so when you wreck the pi bond system, you pretty much ruin things as a pigment, and stuff gets bleached, it gets white. We talked a little bit about deterring insects, maybe, if those experiments with aphid eggs are really indicating something significant and consistent across many different kinds of plants. And now, for a little show from good old California, all from the east side of the Sierras. One of our Gloria colleagues, Adelia, came along this evening, and she's very familiar with this mountain right here, which is Dunderberg, is one of our Alpine survey sites. That peak, and one right down there. And this is one of the prettiest places in California. If you haven't been down 395 in October, between Bridgeport and Conway Summit, this is where that is. Beautiful display of aspens. And if your timing is right, sometimes some sort of lesser, no, I won't say lesser, but little known plants like willows, which can turn a beautiful scarlet and really add to the color. The aspens can be just like flames, tinged with red, brilliant, brilliant yellow. And one of my favorite sights is just aspen leaves against the blue sky. Now, we may not have all those reds and things like that the East Coast has, but we probably have better deep blue skies most of the fall, and so that's pretty good too. Sometimes they have some anthocyanins, and often you see this in clonal groups. Aspens grow in big patches, which are clonally related, and you'll see one patch turn sooner or later than another. You'll see one patch be more yellow than another, or one patch be more tinged with crimson than another. I have no idea why that is either. And kind of the whole array of colors, including the green ones. Don't forget, green is kind of a pretty color as well. And when we see that in contrast with these senescing leaves on the deciduous trees, it just adds to the richness of that color show. And that's all right here in our own home state. And you can find it. So, fruits, that's another part of the plant, and they certainly have their color as well. Lots of different colors. We have cranberries, tomatoes, pumpkins, lemons, rose hips, various kinds of peppers in the eggplant. And I know what that is. That's a dogwood.
Oh.
Yeah. And mandarin. And this one we captured recently. This is a tomato that has a lot of anthocyanins. Most tomatoes don't have any. But in the peel, and we cut one open and took a picture, just starting to color the interior, but when it's ripe, it's all purple. So anthocyanins can be introduced into tomatoes. In this case, they were. So what do the fruit colors do?
Is that indigo rose? That tomato?
Is it what?
Is it indigo rose?
I don't know.
Probably
Might be.
is, yeah.
Yes. So why do plants have pigments in their seeds? Because something eats them and processes them through their gut, which sometimes makes the seeds more readily germinated, and leaves them wherever they leave them, which distributes them. Roots and stems can have pretty good color as well. Beets, obviously. This is bark from white fir and red fir. And this is a plant we took out in our yard, Rosa Sharon, and it had this beautiful yellow pigment in it. Well, why would a plant have a pigment in its roots? Nobody can see it. I don't have any idea. One of the uses of pigments is, if you ever need to tell red fur from white fur, which can get kind of frustrating, well, the needle bends this way, the needle bends that way, just take some of the bark. White fur can have rich browns and lighter colors. Red fur will always have some purple coloration that reminds you of a beet. And I found that to be the most consistent discriminator out there. So just take a little bit of the bark and cut away the gray weathered part and have a look. If it looks kind of the color of a beet, it's a red fur. And it can be dark and chocolate brown, but not purplish, and it's a white fur. So what about those colors? I don't really know. Maybe they're coincidental. Maybe they're antioxidants. Maybe they have something to do with deterring insect attack or herbivory from other things. They can be pleasing to humans, but that's probably not why they evolved that, except in the cultivated ones. So, I'm almost done, Kevin. The uses of plant pigments. There's one, we eat them. This all came out of our garden. Well, the corn didn't. But beets, squash, and greens, all colored with beautiful plant pigments, betalains, carotenoids, and chlorophyll. Very healthy antioxidant molecules, so plant pigments are good to eat. And people have used them forever for various and sundry ways of coloring their fabrics, coloring their bodies. This is annatto, which is a common source of dye. This is Brazil wood. They use the bark as a source of dye. Many, many, many plants have been used for their pigments to color things that humans wanted to make prettier. And we're not the only ones that use them. Animals, birds. This is a goldfinch. You know why it's gold? It has carotenoids. Animals don't synthesize carotenoids at all. So this goldfinch is gold because it got carotenoids it derived from its seed food source and ended up with them in its feathers. Now, this bowerbird also makes use of plant colors and setting up not attractive plumage, but an attractive boudoir there. This set up some lovely colored berries and some other colored berries and some things, I don't know what they are, but plant colors are often, in this case, used as decorate. It's a little bit of bird feng shui. This is... And there's one other really cool thing about how we use plant pigments. This is a nice complicated picture, but it has a very simple message. This is the yellow helices represent a protein that's threaded back and forth through the membrane of a cell. There's the membrane. And right down here, this orange thing is the photosensitive part of the molecules that allow you to see. And this is basically half of a carotenoid, retinol. And that's the basis for vertebrate vision. We derive these from plants. So without the plants that we eat, we wouldn't even have the light sensitive. This is the key thing that changes shape when you perceive light in the back of your eye. And it comes from good old plants. So, maybe one of the benefits is it's just plain pretty. We all enjoy the beautiful aspects of plant colors. And keep in mind, you wouldn't even enjoy these plant colors if you hadn't already eaten some of these plant colors. So they endow us with their beauty and also our ability to see it. So, kind of rambling back over this, what did we find out? What do we know, hopefully, that perhaps we didn't know before? Plant colors are produced largely by pigments. Our explanation, we didn't want to have it stop there. We wanted to look a little more closely at how that works, and hopefully we have. We looked at 4 pigment groups that are responsible: the carotenoids, the flavonoids, the betalains, and the porphyrins in leaves and flowers and fruit. And we looked at the fact that you don't have to change things very much to fine-tune these things and really change the colors that they absorb and therefore the colors that they produce for us to see. Together, that accounts for a lot of the color diversity that we see. When we look at leaves going through their life cycle, we see shifting balance of plant pigments that serve the leaf in various ways, especially in the autumn leaf colors. Light absorption plays many roles for plants, from primary energy production to coloration that protects other components of it, to attracting the interest of pollinators and seed eaters. and we still don't have all the answers. We made quite a tale about how important anthocyanins are to this recovery of nutrients, and then you say, well, what about leaves that don't have them? And nobody knows. And so we don't fully understand all the implications of plant pigments in terms of their ecology. And I would just encourage you, before we end this up, to take a careful look yourself. Be a little bit more attentive to the patterns and the timing and the colors and what's going on in leaves and flowers and fruits, and see if you can figure part of it out for yourself. So I've enjoyed talking about this with you. You've been very patient. I'm not too far behind. And so if we have time, we can maybe have some questions, as long as they're not very hard. Thank you.